So, I welcome you all to my YouTube playlist for AS Biology. And in today's class, we will look on toward understanding one of a very major concept that you people have need to have a grip on, and this is termed as a cytoskeleton. Okay, and once you are done understanding the structure of the cytoskeleton, then it would make it clear for you that how do the centrioles and the centrosomes that we have, and the cilias and the flagellums that we have, so how they would work, because all of these they have something common in them, and that would be the microtubule. If I were to tell you, overall we humans have the muscles and the bones and the tendons and the ligaments, and that is making the structure of the human body. So you see, commonly within the cells we don't have such things, but the cell, it has to form its own cellular skeleton, and that skeleton of the cell is entitled as the cytoskeleton. So we must be aware of that what would be those components that would make up this cytoskeleton. So the names are that it would include the actin filaments, then there would be the intermediate filaments, and at the end we would have these microtubules, and collectively these three things they would be making up this cytoskeleton or the cellular skeleton of the cell. Our approach in terms of the microtubule would be, it should have to be very much clear. So what we know is that these microtubules overall they are long, they would be rigid, and they would appear in the form of the hollow tubes. Okay, so being long, rigid, and being hollow from the inside are the characteristic features of these microtubule. Now, microtubule itself is not the cytoskeleton, but is one of the component of the cytoskeleton. If you were to know about the dimension of these microtubules, so they would be 25 nanometer in diameter. Now, before I teach you that how come these microtubules they would be formed, there is this word that you have to remembering is the word is polymerization. Word polymerization is that if I would take many small repeating units which are monomers, and if these monomers they would begin to combine with one another over and over again, so we would end up making the polymers. So you feel like that this microtubule is a large molecule, this is a polymer, and the monomers that we have got to be making it, so they would be the alpha tubulin and the beta tubulin, and they are proteins with us. Now, let us say that if I have to make one microtubule, then what I am required to see or what I am required to do. So initially I would have this protein, and this protein is the tubulin protein, and this tubulin protein exists in the total of two forms. One of the form of the tubulin is alpha tubulin. The second form of the tubulin is beta tubulin. So when this alpha tubulin and the beta tubulin, when they combine with one another, so they would end up making the diamond molecule. Again, these diamond molecules, they would combine again and again. And as a result of that, we would make a structure out of it, and that would be a protofilament. But if I have to make one microtubule, then total protofilaments that I require to be joining with one another, there has to be a total of 13 protofilaments. So when we would have 13 protofilaments aligned side by side and arranging themselves in the form of a hollow cylindrical structure, so we would end up making one microtubule. So if I were to show you the diagrams here, so you see that here I have got one form of the tubulin protein, there would be alpha tubulin, then we have the beta tubulin, and collectively they have made the dimer molecule. And these dimer, they would combine with one another over and over again, and when they would combine over and over again, so you see that we have made one, you could say, protofilament out of it. Okay, so this is one protofilament that we are making out of it. And when I would combine total of 13 protofilaments, like if I mark it for you, so you see this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 13. So you see that when 13 protofilaments, when they have aligned along one another, so they have made this hollow structure, rigid structure out of it. Now this is one microtubule with us. So overall situation is alpha and beta tubulin making dimer, dimer making protofilament. In 13 protofilament, they would make up one microtubule. Now here is another diagram to make your concept clear at how this is happening. So again, you see I have got the dimer. These dimers, they are combining with one another, and we are making protofilaments. The protofilaments initially they would align side by side, and afterward they would attach with one another from the two ends, and as a result we have made this one microtubule out of it. Now the situation is that we must see in case of the cells that how come this cytoskeleton is assisting the cell, and microtubule was one part of it. Now, as I have said that the intermediate filament and the microtubule and the actin filaments, they all collectively make the cytoskeleton of the cell. If I were to be zooming this, so here you see we have these intermediate filaments here, and then we have got these green ones that here are lying. So they are the microtubules. Okay, and then usually in the muscle cells we have got these actin filaments as well, and together they are making the cytoskeleton of the cell. Now the situation is that how come this cytoskeleton is actually assisting the cell? So as we have done earlier, that we have these mitochondria and we were making the Golgi vesicles and they move around in a cell. So whenever the organelles they have to move around in a cell, so they actually require a media on which they would attach to and then they would move. So what we say is that these organelles, if they have to be moving inside the cell, okay, so it is some kind of an intracellular kind of a movement system that we have got for the organelles, and for that these microtubule they are contributing the role that they allow the Golgi vesicles and the mitochondria to be moving within the cell cytoplasm. So now moving further, here is something that is also related to these microtubules, and we have to be understanding that what centrosome and the centrioles actually are. Now the situation is that these centrosome and the centrioles, they are the unique feature of the animal cell and they would be absent within the plant cell. Now you see that for this animal cell, I have got this cell membrane with me. Yeah, this is one of the part of the protoplasm. I have this nucleus with me, and when I come close to the nucleus, so here I have got this part, and this is the centrosome, and the centrosome on the inside has got these two structures, and they are aligned at a right angle to one another. So this is the centriole with us. So two centrioles at a right angle or at a 90-degree angle to one another, they would make the centrosome, and this lies close to the nucleus. Okay. So let me just quickly tell you about the description of these centrioles, that they are the paired structure. Like this is one centriole, this is another centriole, and they are aligned at a right angle to one another. Now, how this centriole is related to the microtubules, because these are the microtubules actually that would align themselves in a different manner or in a different fashion. And as a result of those microtubules combinations, we would be making these centrioles out. So the overall idea is that these centrioles that we have got close to the nucleus, so they are made up of the microtubules. And earlier we know that the microtubule need to have alpha beta tubule in the dimer, then we need to have protofilament, 13 protofilaments, they would make one microtubule. Okay, so this is how we have to arrange those microtubules further, and uh, at the end we would end up making the centrioles out of it. As far as the dimension of these centrioles are concerned, so they are 500 nanometer long. So what you have to see at the moment is that how come I be making the centriole out of the microtubule? So you see that this is one microtubule, this is second microtubule, this is third microtubule, and overall this becomes a triplet of microtubule. So if I take triplet of nine microtubules, like this is one triplet, this is second triplet, this is third triplet, and likewise, if I keep on arranging the triplet of nine microtubules, then we would make up one centriole. So the usual calculation is that if I have to make one centriole, then how many triplets I require? I require nine triplets, and each triplet has got a total of three microtubules in it. So I can say is that in one centriole, total of the microtubule that we will have with us, say would be 27 microtubule, but these 27 microtubule, they would be arranged in the form of triplets. And triplets of how many microtubules? Nine triplet of microtubules. We need to have to be making one centriole. So if I were to take you to this diagram, so here if I just number it up for you, so here you see that in this diagram, so this is one triplet, okay? And if I go down, so this is second triplet, this is third triplet, this is the fourth triplet, this is fifth triplet, this is sixth with me, and likewise this is seven, this is eighth triplet, and here I have got the ninth triplet, and overall we have made the centriole out of it. Now, moving further, we must be very much clear that what roles these centrioles they would be performing in a cell. So, what we know from our previous classes is that these centrioles they have got a distinctive role in controlling the cell division. Okay, now what do we mean by the cell division is that at any part, one of the parent cell that we have got within our body, so it decides to be making two daughter cells. Okay, and if I were to speak about our cells, so the total of the DNA molecules or the chromosomes that I have got within the nucleus of the cells, so they are 46. You just assume at the moment is that let us just stick to the 46 DNA molecules, okay? And if I have to equally give the 46, you could say DNA molecules or the chromosomes to the daughter cells. So prior to that cell division, I have to duplicate the number of the DNA molecules or the number of chromosomes, and that would be 92. Now, once we have, like initially, if I had this one DNA molecule, and if I have duplicated it up, now when they would be going into the mitosis, so these DNA molecule they would appear in a highly condensed form within a chromosome. Okay, and if we look on toward the diagram of this chromosome, so in this chromosome we have got two chromatids in it. Again, they're termed as the sister chromatids. Now the scenario is that one of the DNA molecule I have to give it to one daughter cell and the other DNA molecule or the chromatid that I have for a chromosome I have to give it to the other daughter cell. So these chromatids they have to be pulled from one another. Now, honestly speaking, I cannot insert my hand into the nucleus where I would be using my fingers to pull these chromatids from one another and I cannot move them to the two daughter cells. So what actually happens is that when we have to do this separation of the chromatids between the daughter cells, so these, you could say, the centrioles that we have got within our cells, so they would make a fiber out of it, and that fiber is referred to as the spindle fiber. Again, the spindle fiber has got the microtubules or the arrangement of the alpha and the beta tubulin, and then further we would make the microtubules out of it, and we are making the spindle fiber out of it. And that spindle fiber would separate the chromatids from one another, and it would pull the equal DNA molecules to the opposite poles of the cells. And then at the end, we would end up making two daughter cells having the same genetic material in them. Now, moving onward, there is another situation. In the airways, you see that you may have learned about the words that there are some ciliated cells, and these ciliated cells, you know, that they have some finger-like projections coming out of them, and they are the cilia. We will see later in another class that these cilia again they are made up of the microtubules. So these microtubules, if they have to be making the cilia, so they need some attachment at the base. So you see that at the base of these cilia that we have got for the ciliated cells, so we have these centrioles lying at their base, and these centrioles that lie at the base of the cilia or the flagellums, so they are again the centrioles with us. So if I were to just simply read it for you people, so what we say is that these centrioles they contribute a role in the cell division, and these centrioles they would make up the mitotic spindle or the spindle fiber, and it would separate the chromatids having the DNA molecules in it. And other than that, we say that these centrioles they would be lying at the base of the cilias and the flagellum, and over there these centrioles they would be entitled as the basal bodies. So I hope you would be clear with the concept of the microtubules and the centrioles and the centrosome. In the next class, we would be looking on toward the structure of the cilia and the flagellum, and again, the base concept would be having a grip on toward the concept of microtubules.